I want to welcome everyone tonight. Thank you very much for coming. My name is Tom Baldwin. I'm the Dean of the College of Natural and Agricultural Sciences. This is the first series of lectures for this year. It's actually the fourth series that we've had in the last couple of years. Before we start, I wanted to recognize a few people. The first group I wanted to recognize are members of our science circle. This is a group of people who believe sufficiently in the mission of the college that they actually support our outreach with their own hard-earned cash. So if members of the science circle, would you please stand or raise your hand or something, and thank you all very much. I also want to recognize one of our science ambassadors, Wayne Kareem, who's back here in the back. The science ambassadors are a group of students within the college who are selected to work on behalf of the college as in events like this, commencement and that kind of thing. Thank you very much, Kareem. I also understand that the superintendent of San Bernardino County Office of Education is here, Gary Thomas. Has Gary made it yet? Well, if I see him walk in the back, I'll try to embarrass him. Finally, let me ask the teachers from the public school system here in the Inland Empire who are with us tonight, would you please stand up and let everyone recognize you, because we really appreciate what you do. There must be more teachers than that. You're going to have to go back and reprimand your colleagues and be sure that they come to the next one. Thank you all very much for coming. Now, one of the reasons when we started this lecture series was to do something about the level of science literacy in the public. And I thought tonight I would just give you an example or two of what I'm talking about. And this slide that has appeared here on the screen is I hope there's no one here from Sprouts. If so, we're going to have a little fun at your expense, I'm afraid. We walked into Sprouts and saw this ad, and it was just priceless. The first thing we asked, $19.99. Yeah, it's $19.99, and the price is not. It's valid through December 31st of '99. But structured water. What would structured water be? What I thought it might be is possibly ice. Okay, so but it's not. So let's see what structured water is. Up here at the top, you can read, it says it's enhanced with electrolytes. At the same time, it's free of chemicals and additives. But here's the good one. Structured water is pure water, perfected using proprietary technology that restructures and energizes the water to become the ideal hydrate. Okay. Yeah, okay. So let's look at another one. This ad, I don't know how many years old it is. Wash away fat and years of age with Lamar reducing soap. The new discovery, results quick and amazing, nothing internal to take. Reduce any part of the body desired without affecting other parts. No dieting. It's all easy, right? This is nothing short of miraculous. I think it must be the kind of lye soap, you know, that your grandmother made. It sort of takes the hide and everything else with it. Okay. Now, here's another one. I lost 12 pounds in 2 weeks. Now, let's do the arithmetic. 1 pound of fat is equivalent to on the order of about 3,500 calories. So if you're going to lose rough basically a pound a day, let's just call it that, you're going to have to consume or burn, or some combination of the two, 3,500 calories more than you usually do. That's going to be a lot of bicycling and no eating. So here's another one. That along the same lines. I lost 20 pounds of stomach fat in just 1 week. I don't know about you, but my eyeball scale says that that's more than 20 pounds. And 20 pounds in 1 week. I think it must have been surgical. But anyway, I think I've made the point. There is so much that you see just everywhere you turn that is just plain ludicrous. And we as voting members of the society, in all seriousness, have a responsibility to help guide the policies of this nation based on good science. So, no more joking around. The lecture series that we have this spring is entitled Science and Society, Major Topics of the 21st Century. Speaker tonight is Dr. Sue Wessler, who has come to us to the Department of Botany and Plant Science. Dr. Wessler is a graduate of the Bronx School of Science. You might notice that I'm not from the Bronx. She was an undergraduate student at State University of New York, Stony Brook, took a PhD in biochemistry from Cornell University, was a postdoc at Carnegie, moved to her first faculty position, University of Georgia. She was there through the ranks. She moved here to UCR just this past year. Sue was elected to the National Academy in 1998, I think it was, yeah, 1998. She was just this past year, she's been elected as the Home Secretary of the National Academy. This is sort of like the Vice President, I guess, of the Academy. Sue's research is in the area of transposon biology. Transposons are sometimes referred to as jumping genes. She is a world's expert in this area. She's also a passionate teacher, and I knew that Sue and I would get along very, very well when she told me about the wonderful job we do of feeding enthusiasm out of our incoming freshmen with freshman courses. There's, I'm sorry to say, some truth to it. Sue is doing some very interesting research, not in the basic science, but in how we teach science as well. I don't know if she'll talk about that tonight, but that's a whole other facet of this interesting person. So welcome, Sue Wessler.
Get some real slides on here.
Sorry about that.
All right. Okay, great. Someone could just turn that light off. Thank you. All right. I'm delighted to be here. New to Riverside, as Tom was saying. I actually live in Claremont, so I have a reverse commute. My daughter is, all my children are here, both my children are here, both my daughters. One is at Scripps College, she's a freshman there, and the other one is at Western University where she's in osteopathy school. So all three of us now moved from Georgia to Southern California, and we are definitely enjoying it. I want to welcome especially the students who came today. And I hope in the future, as Tom was alluding to, and I will mention a little more at the end, have more connections, especially with the community colleges and even the high schools in this area. Because as he said, I think it's recognized nationally that we are doing not a very good job exciting students about science. In fact, we're probably doing the opposite. So tonight what I'm going to talk about is the person the research that I work on, but not the research. I'm going to talk about the historical background, and there'll be a little bit of detail, scientific detail, but mostly just enough so that you can understand why I'm excited about this area. So the first slide, so we can't get that light off, huh? It'll look a lot better if it's off. Okay, the first, okay, that'll be okay, that's fine. So this sort of summarizes a lot of what I'm going to talk about. I'm going to talk about spotted corn kernels, because it actually introduces you to the idea that there really aren't any stupid questions in science, that to ask a question of why are these corn kernels spotted ended up starting a revolution in biology, and that's the story I'm going to tell. And to tell that story, I have to tell you about the scientist who's Barbara McClintock here, and about corn, and about its domestication, and then about the impact of her discoveries. Okay, get my electronics straight here. So as I said, I've divided the talk into 4 parts, and at the end I'm going to tell you a little bit about the new teaching center that has been built at UCR, and just briefly what type of courses we're going to teach there. So the scientific problem is spotted corn kernels. So to explore that problem, I'm going to tell you a little bit more in detail about the plant, which is corn, the scientist Barbara McClintock. Then I'm going to tell you about the solution to the problem and why that solution really led to a brick wall. That is, it didn't go anywhere for a very long time, and I'll explain why. And then finally, why after, I think this is summarized in what you have there, after 40 or 50 years, McClintock finally got recognition for this pretty amazing discovery. And then I'm going to tell you about how that discovery has really led to a revolution in biology, which is ongoing now, in fact accelerating now, and then talk about this dynamic genome courses, which we'll be starting to teach a little bit in the summer and then in the fall. So as I said, this is a little redundant because I was taught you tell them and tell them again and tell them again. So the problem was, as I said, is why are these corn kernels spotted? And many of you around Christmas time, when you go to supermarkets, may see, around Thanksgiving, will see the Indian corn, and a lot of it, if you look closely at it, most of you probably haven't looked closely at it, it has spots and stripes on it, and that's what I'm going to begin with. To do that, I have to tell you about the plant, which is one of the plants that I work with, which is corn, and then about the scientist, Barbara McClintock. So, corn that we eat is generally, in this country at least, colorless. But in most countries in this world, especially in South America where they eat corn, it looks like this. And in fact, if you go, as I said, Thanksgiving, or if you go to health food stores, maybe like Sprouts, but I actually love going to Sprouts. If you go to Sprouts, they'll tell you that this is healthier for you, and it's really not. So the question, I've sort of reduced it as, why are these kernels pigmented? And I've reduced this to a simple biochemistry and genetics lesson, and that is, how do we get, this isn't working, okay, nope, gone. All right, why do we, so to have, this is a pigment, and to make a pigment you need a biochemical pathway. And I've just illustrated that as compound A is converted to compound B to compound C, and compound C then is converted to the pigment. In order to go from A to B, B to C to the pigment, we need enzymes, and as most of you know, enzymes are proteins. Proteins are encoded by genes, okay? And in this case, what I've done is this is a highly variable pointer. It works about 10% of the time. So what I've shown here is, here's a gene that encodes an RNA. That RNA is translated into a protein. That protein is an enzyme, and that's this enzyme here. So there are actually 3 genes that make this pigment. When they're all working, we make pigments, okay? And that's what we show here. This is the normal situation, pigment. So how do we get a colorless kernel? And the colorless kernel is the stuff we eat, right? And we eat mutant corn, all right? It doesn't, and the reason I think that breeders got rid of pigment is because people don't really like eating blue food, and things like that. It's less annoying or something. It's less bothersome if it doesn't have color in it. So they bred the color out of it. And the way they did that is they worked with a gene, and there's lots of genes like this that have if there's a mutation in this gene, you can't make this protein. If you can't make this protein, then you can't convert A to B, B to C, C to the pigment, and you end up with a colorless kernel. Okay, so we got a colorless kernel. All right. So I told you about how we get a pigmented kernel. I told you about how we have a colorless kernel, but how do we get a spotted kernel? All right. So in order to do that, I'm going to backtrack a little bit and tell you about the organism, not really because it has anything to do with the story, but because I think it's kind of a neat story that people don't really think of where did their food come from. And I don't mean farmers' fields. I mean like thousands of years ago. So corn is actually the major cash crop, the major food crop of this country. It's about a $100 billion industry. So it's nice in a way to work. It's also a very good genetic model system. So corn is a domesticated crop, meaning that human beings actually, and this in the case was Native Americans, and I'll mention that a little bit more, started with this plant, which is teosinte. It's a grass, and over really probably hundreds of years, they selected variation in the teosinte that eventually, because of the certain genetic changes that they were selecting for, we ended up with modern corn. And modern corn, without human beings, and this is, I think, one of the interesting things about corn, once we're gone from this planet, corn will be gone within a couple of generations. And the reason is because this ear, so normally what a plant does is it'll scatter its seed. But corn, I mean, you know, it's all the seeds. These are the seeds of corn, obviously. They're all tied up in the husk. And the reason humans selected for that is because it's easy to harvest. Well, it's easy to harvest, but for a normal organism, it's not a way to reproduce. So what will happen is these ears, when humans are gone, you know, the last cornfield, the ears will fall off. All the seed will sort of germinate at once. They'll all strangle, and within, like I said, a couple of generations, corn's gone. But teosinte isn't, because it's a nice, happy wild plant. And this is similar for all our domesticated organisms, whether they be plants or animals. Pigs, I mean, wild pigs don't have different teeth. Domesticated pigs wouldn't survive in the wild for, I'd say, well, not very long at all. Certainly not among wild pigs. Maybe around supermarkets they do fine. But anyway, so the teosinte ear, it's really amazing how much change exists. So this is the same species as maize. So you can cross these plants. They're exactly the same species. But it's just the selection over hundreds and hundreds of years that led this ear, which is a teosinte ear, to evolve to be by human selection. What was selected was an ear that looks like this, and then over the last century, or 2 centuries basically, through improvement, improvement meaning smaller modifications, smaller changes, it went from this ear to the ear that we know and love today. So the difference in size is just unbelievable. And this is an incredible breeding experiment that was conducted about 9,000 or 10,000 years ago. It was conducted in Central America, so actually Mexico. That's where the origin, that's where teosinte is from. What else did I want to say about this? Most of our plants, in fact, most of our plants were not domesticated in North and South America. Most are from Europe, Asia, and Africa. So rice is from both Africa and Asia. Wheat is from Middle East, and I think also from China. So sunflowers are from North America. So there are a few plants that started here, but not that many. Okay, so corn was the reason why the Incas and the Aztecs became successful civilizations. Oh, okay, thank you. Okay. So people were hunter-gatherers. So they could not build a civilization. They couldn't stay in one place. So crop plants allowed them to plant a significant amount of food, harvest it, and then store it over the winter. So this bounty of food allowed people to go from being just farmers and hunters and gatherers, so to speak, to be artists, writers, whatever. So it really allowed for the growth of modern civilization, the development of modern civilization. And this is reflected in a lot of the artifacts from Central America. So if in working on corn, anytime I go to like New Mexico or places like that, I could always get nice earrings and things because it really it's part of their it's a common theme in their jewelry and in their icons. All right. So the tremendous diversity of corn is due in large part, not large part, but a significant part of it has to do with a very dynamic genome that corn has, and that's what I'm going to start to tell you about now. So we go from the plant to the other part of it, and that's the scientist, who is Barbara McClintock. So I had the incredible good fortune to know this person who became a woman for about the last 10 years of her life. She was a very unusual child. She was born in Hartford, Connecticut in 1902. In 1908, her family moved to Brooklyn, New York, so we're both New Yorkers. She got her bachelor's degree at Cornell in 1923. So I say she was an unusual child because back in the early 1900s, children didn't have to go to school. There were no laws that said you had to. And she was kind of bored with school. And so her parents said she could stay home. And she was basically homeschooled. She was also very unusual in that she was sort of a tomboy. So she liked to climb trees. She liked to do things like that. And at that time, girls didn't wear pants. And so you couldn't go to the store and buy pants. So her mother would sew her knickers, and you'll see that in a picture. Every time I've seen her, every time I saw her, she was always wearing pants. She was, I'll say in a second, a little more here. All right. So after she got her bachelor's degree at Cornell, she had a problem because virtually no graduate programs in the country in the sciences accepted women into the program. So one program that did was at Cornell also, and it was a genetics program, and it was R.A. Emerson, and that's him here. This is Barbara in knickers, okay? And this is in front of a shed in the Cornell plantation, which is still there to this day, with a plaque on it commemorating this group. So this was in 1929 in Ithaca. And so corn before World War II was one of the two model organisms, the other being Drosophila. Okay. So McClintock was a cytogeneticist. This was sort of like the genomics person, the person who looked at the whole genome back then, before we were able to sequence genomes and things like that. And what was done is you would take cells and you would stain them so that you could see the DNA. In this case, what you're seeing, I'm not sure if it looks like meiosis, could be mitosis. Natasha, do you know? No, one or the other. Anyway, these are the chromosomes, and here you could watch the whole genome, and you could learn a lot. Excuse me, somebody's there. Oh, okay. So that's what McClintock did, and she was the finest cytogeneticist in the world. She made amazing discoveries, and she could not find an academic position. And this is because women in science at that time either were teachers or they worked in the labs of their husbands. And she essentially, what I did mention before is her mother did not want her to go to college because she said college would make her unmarriageable, which actually turned out to be the case. She never got married. But so she didn't have a husband whose lab she could work in, but she had a lot of really good friends, and she ended up finally getting a job at the University of Missouri, and that's a picture over here. She then went from there. It didn't work out. She wanted to do a research. She did not want to be on committees. She didn't want to teach. She didn't want to do all the things that faculty members are supposed to do. She only wanted to do research. And so she ended up going from the University of Missouri to Cold Spring Harbor, which was then the Carnegie Institute at that time. And she worked there from the 1930s, late 1930s, until her death in 1992. And this is a picture of her in front of her microscope, okay? Which I had the, as I said, great privilege to visit her and look through that microscope. Okay, so this is a picture of her. So there's lots of myths that are out there about McClintock, that she was kind of a recluse, she was a loner, that she was this kind of intuitive genius and all this kind of stuff, and that people didn't believe her. that she had these really crazy ideas, not crazy ideas, but ideas that people, and that just is simply not the case. McClintock was recognized in her time. She was elected to the National Academy of Science in 1944 at the age of 42, which is very, very young. She was only the third woman elected to the National Academy of Science, and I should point out that of the 3 women, 2 of them went on to win Nobel Prizes. So if you think the bar was a little higher, I think so. So she then, in 1945, she was elected president of the Genetic Society, and this really is a measure of how well known she was in her community. So the idea of her being a recluse is not really true. So we've gotten through McClintock. So the next thing is the problem, these spotted corn kernels, and what was her solution to that problem. So I've divided that into several parts, 3 parts. First is, what was her solution? The second was, why did a solution really take so long to sort of permeate throughout the scientific community? And finally, when it finally permeated, the recognition was significant. So again, why are these corn kernels spotted? And I said before that I told you why we have pigmented kernels, because we have a pigment pathway and all the genes are working. I told you that there are mutant kernels, that if one gene is mutant, and what I didn't mention was that a gene is mutant, as many of you know, if there's a change in a codon, if there's a change in the DNA sequence, if there's a deletion. Those are mutations that we're all familiar with, and those are the ones I'm talking about. But for spotted kernels, we needed something else. So this is a picture, a page, a scan of a page from McClintock's notebook. And it really shows she was a geneticist, so she performed crosses, and through those crosses she figured out what was going on in kernels that were incredibly, very high resolution in terms of their phenotypes, as we call it, what they look like, and what is going on at the level of the DNA. And I'm not going to discuss this in detail, but essentially what she determined was she determined that the spotted kernels were caused by a new class of mutation. It wasn't a deletion, it wasn't insertion, it wasn't a deletion, it wasn't a base change. It was actually, here's our gene, and it's a piece of DNA which she called a transposable element or controlling element, and it inserts in the gene, but unlike most mutations, this has a special property in that the element, once it goes in, it can come out. So it could either be in or out. So it's as we say, it's a reversible mutation, okay? So let's go back and let's look at our corn kernels here. So what we have here is in these sectors of the colorless sectors, we in fact have the transposable element stuck in the gene. And when it's stuck in the gene, we can't make the pigment, so it's colorless. But as the kernel develops in certain cells, the transposable element can jump out. It can excise, doesn't jump. And once it comes out, it restores the expression of the gene, and we end up with these sectors. Okay, and that's basically it. So that's, I mean, it seems pretty trivial, right? Okay, now I'm going to tell you why it's not. Like I said, the brick wall really was, as I show here, that McClintock discovered transposable elements in the 1940s. Okay, she received her Nobel Prize 40 years later. Now, Nobel Prizes are usually awarded at least 10 years after the discovery. 40 years is a very, very long time. The only time that's really longer, I think, was Peyton Rouse, who discovered Rouse sarcoma virus, I think, in 1912, and he received the Nobel Prize almost 50 or 60 years later. One thing you have to do is live a long life, because they don't give posthumous Nobel Prizes, okay? They're not given out posthumously. So what it really was with McClintock is that she was really ahead of her time. It took a long time for the scientific world to recognize the importance of these strange pieces of DNA that she found, okay? And when they finally recognized it, she received the Nobel Prize. So why did it take so long? Okay, it took so long because after her discovery in maize, in corn, so people thought, oh, these strange things are just in corn. And corn's a domesticated plant, so something must have happened during domestication that caused these strange genetic elements to pop in or pop out or whatever, and it really isn't true for the rest of life. But over the years, people found that, first of all, transposable elements were discovered in the 1950s in Drosophila, the fruit fly, which is the other model genetic organism. In the 1960s, they were discovered in bacteria, in E. coli. And finally, the most important discovery, in the 1970s, they were found in the human genome. And once they were found in the human genome, and found to be very abundant in the human genome, then she was sort of eligible for a Nobel Prize in medicine or physiology, which means that your discovery is relevant to human health. Okay? So this is a picture of, just to show you how, if you go out and look, and we have Jim Burnett, who I'll show a picture of in a second, in our class, we ask students to look around their environment, mostly flowers, for evidence of transposable elements, and take an iPhone picture of it or something like that. And if they can do this, they'll get extra credit. So this is a picture from actually in Napa. We were in a winery in Napa, and this was there. And then one of my colleagues working on petunia sent me this just gorgeous picture, which we ended up putting on the cover of our textbook, of a petunia flower. And what you see are these pigment sectors are exactly like the spots on the corn kernels. Okay. And you can see these in lots of ornamental, a lot of ornamental flowers. Azaleas, there's a lot of azalea strains that have transposable elements. Okay, recognition, this is actually a stamp. So you can see Barbara made it big. She's one of the few geneticists that were on a U.S. stamp. So what was really the importance of her discovery? Okay, so what she discovered was a component of chromosomes. So the chromosomes, as we know, so our genome, I should really define this, our genome are all the genes, all the genetic material that's in each of our cells. Humans have, I work on plants, so if I don't know this, 23 pairs of, 22 pairs, something. We have lots of chromosomes. Corn has 10, I know that. And so before McClintock, find this again, people thought that our chromosomes were made up of genes, and exclusively of genes that are lined up, so that each of these blocks would be a gene. So gene, 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 like beads on a string. But in fact, what McClintock discovered, that in addition to the genes, there are actually these mobile DNA elements, these transposable elements, that reside in the genome and that can move around. Okay? What wasn't known until fairly very recently is just how much of our genomes and the genomes of life on this planet arise from transposable elements, and that's what I'm going to tell you about now. But for this discovery in 1983, she was awarded the Nobel Prize in Medicine or Physiology. These are usually awarded to up to 3 people. The fact that it's very unusual for someone to be awarded this alone, as a single person. The other thing that of significance, sort of selfish significance, 1983 was the year that I started as a faculty member at the University of Georgia. And all I can tell you is there's nothing better to happen to you is to start as a faculty member working on something that then has received the Nobel Prize. So everybody invites you to give talks and all that, because Barbara wasn't going around giving talks. So that was a real fortuitous beginning. Okay. And so like I said, I knew her from 1980 until her death in 1992, and just, I mean, I just consider it certainly one of the great things that ever happened to me is that I got to meet this amazing person. Anyway, so the revolution of biology. All right, so I'm just briefly going to tell you what are transposable elements, and when I say briefly, I mean really briefly. But I'm going to tell you a lot about how abundant they are, just how much of our genomes, our chromosomes, are transposable elements, and then what good are they? All right. So this is one of 2 sort of technical slides. Transposable elements are pieces of DNA. There are 2 general classes. This would be DNA. They usually encode a single protein. Then that protein allows them to move around. So they really are a world unto themselves. And it's the fact that they can move around and increase their copy number that they survive. Okay. So there are 2 types of elements. The other is called a retro element, and in fact these elements are very, very similar to retroviruses like the AIDS virus, But unlike the AIDS virus, and I'll show you in a second, this is the last technical slide, I promise you. So this is a piece of DNA here, and that would be a retrotransposon. The way they move around, retrotransposons, is we make a transcript, an RNA. That RNA is then turned into, through enzymes encoded by the element itself, a double-stranded DNA, and that DNA then inserts someplace else in the genome. So what you have is like a printing press. You have an element that can churn out lots and lots of RNAs. Those RNAs can be converted into DNA, and those DNAs go into the genome someplace else. So you can end up with, there are some retrotransposons in some genomes. The human genome has 1.5 million copies of a single retrotransposon called ALU. Okay, it's 10% of our genome. Okay, it's this little transposable element. So these guys are really successful. All right, so how abundant are they? I just told you a little bit about that. So my favorite organisms and the organisms that are critical to life, human life on this planet, are the cereals, which account for 60% of human calories on this planet. And so we have rice, sorghum, maize, barley, wheat, and oats. So, you know, I'm from the Bronx, as I said, and these all look pretty similar to me. In fact, they're all grasses. You know, they look like grasses when they grow, all right? But what is really astounding is now that we have sequenced many of these genomes and determined the size of them, you see the genome sizes. So rice, and I'm going to show you, rice genome size is 450 million base pairs, and I've abbreviated that MB. So sorghum, which again has 700 million base pairs. Maize is 250 million base pairs. This is the size of the human genome. Okay? Barley, 5,000 million base pairs, 5 billion base pairs. Wheat is 20,000 million base pairs, and oat is off the scale. These organisms can double their genome. So there's two reasons why their genomes are so big. One is because they actually can form what's called polyploids, but the main reason is because, and I'll show you this in a second, this explains. So all of these cereals, let me go back in a second, and we know this now because these genomes, certainly of rice, sorghum, maize, barley, have been sequenced either to a certain extent or completely, and we know a lot about the wheat genome. They all have pretty much the same number of genes, about 30,000 genes, okay? So despite that, the genome of rice is 1/6 the size of the genome of maize, and, you know, less than 1/10 the size of barley, but they all have the same number of genes, pretty much the same number of genes. Why? What's different? It's basically how much the transposable elements, it's due to transposable elements, that's the bottom line. So here we take the smallest genome, rice, all right? Here's I've drawn this as 3 genes that are sitting in a rice chromosome. In maize has those 3 genes also, but they're further apart from each other. Barley has those 3 genes, but they're even further apart. So what's happening is these elements are not going into the genes. They're just sort of pushing the genes apart. Okay? Weird. All right, and this gives you an idea. Here is a region of the barley genome that a colleague of mine, Alan Schulman, determined. So here's a piece of DNA, and all of these things there are transposable elements. And here are the 3 genes. So in this region of maybe 100,000 base pairs, maybe 10,000 are genes, and 90,000 are transposable elements. Okay? All right. So let's say, and I think this is in the handout there, so we can do this quickly, what fraction, I do this with, when I used to teach introductory biology, I would say this, you know, what fraction of the human genome is derived from transposable elements, and the answer is D. Over 50% of the human genome are transposable elements. Okay? So, and this is the way I illustrate that to the students in the class is to say that our typical introductory biology textbook, is 1,000 pages. So if every letter in the textbook was in fact a base in DNA, A, G, C, or T, there's 2.5 or 3 billion bases in the human genome. If we had like 12-point fonts or something, that would take up, without pictures, without things like that, would take up about 1,000 volumes of 1,000 pages each, would be our entire genome. Okay. Of that, 50 volumes would be our genes. That's it. 50 volumes out of 1,000. 500 volumes would be transposable elements. So that's what I say here. It's equivalent. 20 to 40 volumes are genes, and 500 of the 1,000 are transposable elements. This was a huge surprise that really started to come out in the 1980s. And in fact, now when they say they're sequencing genomes, most of what they sequence are transposable elements. And the thing is to find the genes. Where are the genes? Okay. And I'm interested in the transposable elements, so it's great. Okay. So our genome is not unusual. So 50% of the genomes we're not alone, our primate colleagues and our mammalian, humans, chimps, mouse, ape, all about 50%. Okay. Maize genome is 75, it's probably 80% transposable elements. Barley genome is 85% transposable elements, and my favorite, beautiful iris, 98% of the genome are transposable elements. 2% of the genome are genes. Okay? And all of these organisms pretty much have 30,000 genes, 20 to 30,000 genes. Okay? And iris, they're beautiful plants. I mean, you could never tell by looking at these things what sort of lurks in their genomes. All right. It's really been a huge surprise and a huge headache for people sequencing genomes. So how do organisms live with so many transposable elements? And this is what I really work on. This is what many people working on transposable elements. The question is, how can organisms survive with this much stuff that really doesn't seem to do anything but maybe bad stuff? Okay. So it turns out that now that we sequence genomes, we can see that most of the transposable elements are dead, meaning they've accumulated lots of mutations and they no longer function. So they're sort of relics. And they're there because it's really hard for organisms to get rid of them. So they're just there. That's one thing. The other thing is that the ones that are active, when they move around, they go to places where they'll cause the least harm. And the way they've evolved this. So you might imagine if there's a transposable element that starts making copies and the copies insert into genes, that organism isn't going to be around to reproduce, or the progeny, the organism's around. The progeny are going to be defective because they're going to have all these transposable elements in their genes. So a successful element has to evolve places where it can go that it will not harm the host. And so it's a wonderful thing to study, the logic behind how these things succeed. The other thing is something that actually is a very popular research topic in the genomics department at UC Riverside, is the host, us, and all living things have evolved mechanisms, called epigenetic mechanisms, that silence, that prevent the elements from moving around. Okay? And probably these mechanisms, it's the hottest area of biology now, I would say, of molecular biology. Is figuring out how epigenetic mechanisms work, and there's epigenetic mechanisms that are involved, and I really don't want to go into it, but they're involved in cancer, they're all the hot topics. But this mechanism probably evolved so organisms can inactivate their transposable elements. So it's sort of like a cat and mouse game. We have these elements that can move around, and we have mechanisms to keep them repressed, but they keep evolving ways to sort of elude those mechanisms. And this is going on, has gone on. All right, so just to give you an idea, a typical human gene here, typical in quotation marks, the typical human gene is composed of exons, and this is, again, one of the last technical slides. These are actually the coding regions. This was also a real big surprise in the late 1970s, is that our genes are not nicely lined up into a nice coding sequence. They're separated by all these things that we now know to be introns. So this is a gene, and most of the gene is actually this dead space, which is not really dead space, which has to be cut out in order to make a good protein. It turns out that transposable elements, when we sequence them, you find them in all of these introns and regions that are not the exons. So some human genes can have over 100 transposable elements in their introns. Okay? So that's an example of a transposable element that goes someplace that it won't affect what the gene is producing, the product. And it is successful. Okay, there's actually lots of different colors mean there's lots of different types of transposons, but that really doesn't matter. This is an example of, this is a piece of the genome of a plant, and here are the genes, and this is sort of what I showed you before, but a different drawing. Here are the genes, here are all the transposable elements that are between the genes, okay? But in fact, in plants, and it's what we work on in my lab, that we have a gene, so I've taken this gene and broken it up into exons and introns, which in fact what its structure is in an organism. And in fact, transposons find places, like I said, here they're sticking in the introns, and these little transposons are there so that when you then make RNA and you look at the RNA, they're removed. So they're there, they survive, but they're not really impacting the host. Okay? So this is how an organism can survive, do quite well, even though it has this huge load of this stuff. I've resisted calling it junk because people always call it junk, and we know it's not. I mean, a lot of it is junk. But anyway, so that's the next question is are they useful? And McClintock, who, I mean, it's very humbling to work in a field where she started the field, because she didn't just discover transposable elements. In fact, her discovery of transposable elements isn't what really excited her. What really excited her is she thought that these were important. They had an important role in evolution. And her theory, which is called the genome shock hypothesis, goes like this, that transposable elements are in genomes, and they usually do not move around. Okay? That stresses of some sort, and we could think of such stress, it's not, you know, in like traffic on the 10 or the 210. It's climate change, some real serious stress to life forms. They could possibly activate transposable elements. So there are a few transposable elements in the genome that are capable of moving around. The host keeps them repressed, but environmental conditions may cause them to start moving around, may activate them. Okay? That this increased movement, in fact, increases the mutation frequency, and what it does is it generates diversity, genetic diversity. So you have a population that's of rice plants, which is what I work on, and we have rice plants. We discovered, us and Japanese collaborators discovered strains within the last 10 years where there's a transposable element that's moving around like crazy. It's inserting in 40 new places in the genome every generation, and it's just diversifying this population incredibly. And the population goes on; it's very healthy. So this was, and that was, so McClintock hypothesized this in the 50s. Okay, we're gone. So the idea here is that the increased genetic diversity generated by transposable elements may allow a few individuals in the population with the right mutations to adapt to the new environment. Okay? So what sort of things can transposable elements do? And this, again, this is 2 quick slides here. Transposable, so if this is a gene, and this would be a wild-type gene, a transposable element could insert near the gene and cause that gene to be expressed differently. So this is a way that the transposable element can create diversity that could be meaningful. One interesting thing found by Evan Eichler, who's at the University of Washington, these are this is the human genome, 22 chromosomes and the X and Y chromosome. And what he found is that about 5 or 6% of the genome consists of duplicated regions. So pieces of the genome are duplicated. So you'll have like 7 or 8 genes over in one place, and that piece of DNA is duplicated like on chromosome 5 when it was on chromosome 2 before. In fact, what he has found is that a lot of those duplications are probably caused by the activity of transposable elements. So transposable elements are generating regions of DNA, duplicating regions, and what this does is it allows the species to maybe evolve new functions. So again, it's a way that to diversify populations, possibly to make them will create diversity where they could adapt to changing environment. And the way that I put it in a grant proposal many years ago is that transposable elements shake up an otherwise conservative genome in ways that we are just beginning to understand. So what I want to end here is to tell you about the course a little bit about the course, but a little bit more about the facility that is one of the reasons that I was hired at Riverside, and that is to teach this course. And I'll tell you to start with, this program started with a proposal that I wrote in 2006 in response to this call from proposals from the Howard Hughes Medical Institute. And I think many of you have heard about Howard Hughes. For the students that are there, Howard Hughes was the aviator in the movie. He was, when I was younger, the richest man alive, and he lived on the top floor of a hotel in Las Vegas, and he never cut his fingernails. Anyway, but his estate, he didn't pay his taxes, essentially, for many years, and so his estate went to fund the Howard Hughes Medical Institute, which has a huge impact on basic science in this country. They have an endowment of about $15 billion, and they fund biomedical research. But they decided in '02, actually, that the science education in this country needs to be improved. And what they thought was happening was that there was a great deal of excitement going on in our research labs, while our classrooms were less than exciting, would be a nice way to say it. And the reason really is that we weren't doing science. I mean, what we love as scientists is not to sort of pontificate about it and to put up PowerPoints of figures in textbooks. We like to do it. That's what's exciting. I mean, it's like sports. I mean, it's participatory. So what they did is they started this. They had a grant program, and it says here a program designed to stimulate personalized undergraduate education by accomplished research scientists. So essentially, they asked for proposals. It was 5 pages you had to write of blue sky, no budget, because if they liked what you did, they would give you your program $1 million over 4 years. And for teaching, that's a lot of money. In fact, I was awarded one in '06, and the grant ended in 2010, but I still have about half the grant, which I've been able to take with me here. So it's like I said, it's hard to spend money on that much money on teaching. So my project really was to design and have built a facility that was like my research lab. So what I wanted to do was create a research classroom that was a copy of my research lab, so that the students could be involved in discovery-based research in something that I knew something about, the research in my lab. And the people in my lab, the graduate students and postdoctorals, could also be involved in teaching. Something that we do, which is not usually done in undergraduate classes, is we focus on a topic. And in this case, obviously, we focus on transposable elements. And the reason I decided to do this is because I taught introductory biology for 9 years at the University of Georgia. And the problem with teaching introductory biology, and I also write a genetics textbook, and the problem with it is the knowledge in biology is increasing exponentially. And so the books are getting bigger and bigger. I mean, I see my daughter walk around with her backpack and, 1,200-page books. And it's not like the students are learning anymore. I mean, what's happening is that we're just drowning. They're drowning in facts. The concepts are disappearing. They're disappearing in all this data. And so what I would see people in my lab, I would see an undergraduate start in my lab, and they would sit in front of a computer, and they would get involved in doing an experiment, and then they would ask questions so they would start with the experiment, and then ask the questions, well, what's this stuff? And they would just go online, or they would go to resources. So you start with a question. You have a reason to care about all of this data. So the idea here is students will focus first on transposable elements, and we use transposable elements, as it says here, as a vehicle to teach evolutionary concepts. And in California, it's not nearly as important as it was in Georgia. In Georgia, the vast majority of the state does not believe in evolution. And so the idea here is that we use it, it's plants, it's innocuous, and you sort of get them to do the experiments, and then they start asking questions, and it's like, oh, it's evolution. It's like, oh, okay. And so that's part of it. And I mean, many of the students in our undergraduate class, our freshman class, even the upper classes, when we asked them what they learned about evolution in high school, they said nothing. They didn't learn about it. And in fact, in some cases, when the high school teacher taught it to them. There was an administrator standing behind them to make sure that they were told about, the other theories. So this was really important to us, to give students hands-on experience with evolutionary research. And the other thing that was really important to us is to have students care about plants. So plants are usually the first thing plant topics are the first thing that goes if there's not enough time in introductory biology. If you ask anybody if there's a picture of a field and a deer in the middle, you say, what's a field, beautiful trees, and you say, what's in the picture, they'll say a deer. And then if there's no deer, they'll say nothing. So it really, and especially now, plants are truly, important to the survival of not just us, but the rest of this planet. And it really is important that people understand. And not only that, we know now, with DNA sequence and everything, that, you could take plant genes and put them in animals. You could take animal genes and put them in plants. It's universal. So you can actually do a lot of experiments, and undergraduates can do a lot of experiments without any safety, the sort of safety concerns you have working with animals. And it's also, if you don't like pipetting thing, it's a plant, so people don't mind grinding it up and stuff like that, because, grinding up spinach leaves or something like that. So, all right, so this is, the main reason I'm putting this slide in is to introduce Jim Burnett, who's in the audience. He was also hired at Riverside to run this teaching program. Jim actually started in my lab as an undergraduate in 1995. Jim, where are you? I'm going to embarrass you. Can you stand up somewhere? All right, there he is. All right. So Jim started as an undergraduate in my lab in 1995, and he went on to get his PhD and then do postdoctoral work, and then really decided that he would much rather be involved in science teaching, and especially teaching research. So these are some of the students in our first freshman class at the University of Georgia. So the course is very simply, it's divided into two halves, all the courses that we teach, which we call the dynamic genome courses. The first half is where students learn the tools, and the tools are both computational and experimental. The second half, they apply the tools to a project. And the projects usually come out of my research lab. They're simple. They have never been done before. So it really is discovery-based, and it can be done within a 4-week period. All right, so what we found works is, like I said, to keep things simple. There's only about 2 more slides. We focus on one topic, as I said, transposable elements. So this is where it's very different than a lot of high school or college lab classes. I just remember, my chemistry lab where, you get to the end and you got this brown powder and everybody else has a white powder, and it's like, why? It's like, well, sorry, and you just go on. Next week we'll be doing something else. And so it's just, it's not inspiring. So here we focus on a topic, a biological topic, which we're all enthusiastic about because we all work on it in the lab also. We keep the methodology simple, okay? So we just focus on a couple of techniques, and we use them to answer questions, and they'll be used in different order, much like putting a sentence together with words. So, and the most important thing we found is students get to repeat failed experiments. And this is all what goes on in the lab. In the lab, you get to do things over and over again until you get it to work. So students get to master techniques, and they really, and we find at least of the last 3 years of teaching it, that they really enjoy success. Students, we have them meet with practicing scientists. We have visitors that come to the classroom. We do Skype interviews with some of the famous people that I know in science, famous in my small pool, not in the real world. And we have a social atmosphere in the lab. So we have coffee, we have music, all the things that are in a normal research lab, and we have breaks where we sit down and talk about things. So I'm going to show you what the lab looks like because it was just built at Riverside. This is not what it looks like. This was the model. So what you see is this is a computational lab at Cold Spring Harbor at the Dolan Learning Center. And I've been there a couple of times, and Dave Miklos, who's the older person sitting there, has run this since 1988. It's mostly for high school students, and he has them come in and do experiments and learn about bioinformatics. And I asked him, what's the best model for a computational classroom? And he said, this is it. So we through the help of certainly Dean Baldwin and the other people in the administration, we were able to build actually what I think is a lot nicer than what they have at Cold Spring Harbor, and it's a two-tiered. These are all going to be. We're buying laptops. Students are going to use laptops. Those are each of their stations. That's a smart board there, where we can project, not just project, but it's basically a computer monitor. It's a touch screen, so we can use a stylus and bring down menus and all that. So this is one room. The other room is a wet lab. So unlike most classroom laboratories, this has both a computational and experimental component where students go back and forth. So here's our version of that wet lab. And we have it organized in these benches. So again, just as the Dolan lab had, students can work together in teams of 4. So this is actually the space where it's at, and it's a building next to genomics. So here would be the wet lab. Here's the computer lab. Here, and I think I show it here, here is a large space for offices and where we can get together, we can eat over here. We'll have a refrigerator here. And this is an open space for hopefully future expansion, or where students, it wasn't demolished, so students can come in after hours and do their projects in this lab. And I am pleased to announce that it's the first floor of the University Laboratory building. It's now the Neil A. Campbell Science Learning Laboratory, as Rochelle Campbell donated the money for the renovations of the space, and we are more than a little proud to name it after Neil and all that he's done for biology education in the world, really. And I think there's going to be a dedication later in the year. So this was, as you can see, we don't even have the chairs yet. This was just really finished. It's not even finished. It's really close. So anyway, I think I'll stop there and thank the people that have, not the people, the funding agencies. There's tons of people that I thank, but I'm not going to do that now. Howard Hughes Medical Institute, NSF, which has funded our program very nicely over the last couple of years. We actually have a grant now with the Keck Foundation, WM Keck Foundation, to look at transposable elements in mosquito, and this is with Peter Atkinson, who's in the back, who's a mosquito transposon expert, and Jason Steach, I don't know if Jason's here, who's a bioinformatics expert, and certainly lots of assistance from Riverside, lots of financial assistance from Riverside in getting the lab set up. So I'll stop there and entertain questions. The mics are in the front if anybody wants to. All right.
Don't be shy. Ask for a question, Mike.
No. It's okay. So there are a couple there. It's not working. Yeah, there you look.
I was going to ask you, how well are you able to manipulate these transposable elements in the lab, and what does that kind of manipulation result in?
That would be another seminar. We can take the elements out from one organism and put it in another, and get them to move, and they move around. So we could take rice elements and put them in yeast, and they'll move around in yeast. So it's pretty universal, the machinery. So that's a short answer.
Yes, I'm trying to get a feeling of how important transposable elements are for survival of species. Are there many species that are known that have relatively small percentage of transposable elements, maybe 25% or less? Is this something that is seen in some organisms, or do most of them have 50% or even more,
Yeah.
like the examples you've given?
Good question. It varies tremendously. So virtually all multicellular organisms, if not all, have transposable elements. and it can vary from a few percent up to 98%. So some plants like Arabidopsis, which is the model plant, it's a weed, mustard weed, it's about 15% of its genome are transposable elements. Drosophila is about, what is it, Peter, about 15%? 4%. Okay, 4%. So it varies tremendously. And we have no idea why. We don't have any idea whether, so one of the models is that maybe organisms that have lots of active transposable elements maybe can evolve more rapidly. We don't know. The types of elements that are present in different genomes vary dramatically. So yeah, and we're just finding this out as we sequence all these genomes. And within 10 years, I would say we'll know what every genome, just about every species, every significant species on this planet, what their genomes look like. Even less time than that. Right. Yes?
Are corn kernels pigment?
What's the color is pigment. Yeah. Yeah. The corn kernels are seeds. Right, and the colors are pigments.
Okay, well, and when you put in the lab, when you put the corn kernels, what kind of elements do you mix it with?
We don't mix it. We just, what we do is we, go ahead, keep going.
Like to make it, like, not fermenting anymore?
We don't do, no, it's just, they're different strains. So if I take this kernel and plant it, okay, if I plant it and grow up a corn plant and then pollinate it and look at the ear, they'll all be yellow. If I take this guy and plant it and grow it up and pollinate it, all of the kernels will be purple. So it's just genetic. If I take this guy and plant it, all the kernels will be, most of them will be spotted. Okay?
Is there any hope of kind of expanding what you learn in your teaching lab to the wider biology sciences and UCR?
Well, actually, our course is going to be an alternative lab course in the introductory series. So at Georgia, our course was sort of laid on top of the existing courses, and we had a really hard time recruiting students because the advisors were advising them not to take the course. So here we've done the work beforehand. We've worked with Rich Cardullo and people, people involved in curriculum to make sure that this will count as an alternative form. And yes, we're going to have other professors who want to teach their specialty, who want to bring their research to the undergraduate classroom, develop classes. That's our hope, to develop classes for this facility. So this facility will not just be used for our classes. It'll be used really for anybody, until it gets a little too crowded. Then we're going to have to just go to the dean and say, build another one. But the types of classes that are being built, the types of facilities for introductory biology are changing dramatically. There's a really good thing, the University of Minnesota just built their new science building. And instead of lecture halls that are very much like this, with the warm personality of this room, ceilings might be a little higher, but they're in the round. They're tables that have like 9 people sitting around, and there is no professor at the front of the room. And they just discuss various problems, and there are flat screens where they can interact with their computers to answer questions and solve problems. So there really is a revolution that's beginning in how we teach science in this country. And the problem is that this stuff costs money, and money is not something that education budgets are being cut left and right. So that's why the HHMI, the private guys with the bucks, Yates Foundation, are starting to spend a lot more on education because it has to be done. So. Yes.
Do we know the origin of transposable elements?
No. They're in bacteria. They were in the earliest life. They're in the simplest life forms. So we think that they sort of came along with life.
So like it's intrinsic to the organism itself, made by it not coming in as something invasive?
Oh, it's definitely part of it. It's inherited from generation to generation. No, it's part of the genome. Every single one of us in this room has 1.5 million copies of this particular alu element. And every one of those copies, except 100, are in exactly the same position in every one of our genomes.
Even farther back, like evolutionarily, could it be something that, you know, at the origin came from?
Oh, no, these are things that clearly inserted, you know, but evolution is a really long time. So if you look at, say, the chimp and humans, we probably share, I don't know what it is, like 50%? 50% of our transpose, 99% of the coding sequences, meaning the protein coding sequences. When you look at the rest of the genome, there's not a whole lot that's that similar. So that's kind of the genes themselves are very similar, but the transposons are very different, can be different. We share probably about 50%, I'd say, chimps and humans of our transposons. It might be more than that, I don't know. I work on plants.
You said something about one other study of stress or something with the DS and that.
Yeah.
Is there, can you talk about that a little more?
Okay. So someone just said before that how do we know that this is intrinsic to the organism and not like a virus infects and puts these things in? So what McClintock would do and other geneticists is you could take a corn, you know, a corn plant, and you could subject it, you could mutagenize it, you subject it to a particular chemical. And then when you plant it and look at the seeds, you find that transposons have become activated. So they are things that are in the genome. They're part of the genetic material, but generally the host, the corn plant, is keeping it repressed. And the mutagen, it's believed what the mutagen does is it causes mutations that then release the repression. So that's how she knew that these were not, you know, from outer space, that they were actually part of the genetic material of the organism. Right. And now we know it because we sequence genomes, and it's most of the genetic material. Right. Yes.
Okay, back to the subject of teaching.
Of what?
To the subject of teaching.
Okay.
First of all, I wholeheartedly agree with your
and you know a lot about this, I know that.
Your plea should be to increase students' awareness about plants and stimulate the curiosity about plants. There's something I've been trying to solve for the past 20 years, and it's not working as well as it might be. Second of all, how practical do you think it would be to scale up your teaching approach to a class the size of 1,050 like we have in Bio 5A?
We can't have classes of 1,050. That's what it comes down to. I mean, we can't. And no, you can't scale it that much. I mean, how big it can be, I don't know. That's what we're going to try to find out over the next couple of years, and that's what's being replicated around the country. How much can you scale this up? You can scale up the computational part. So you can have students do really exciting experiments, you know, online, essentially. But, you know, when we have the students do computational stuff in the wet lab, they would much rather be in the wet lab than in the computer room. So software, the programmers and people who write software, you know, it's a big challenge to try to develop engaging programs. Because most of the computer programs that are used in the lab are sort of geeky. I mean, they're written for scientists. And so they're not fancy. In fact, they're really difficult to use. And so we've been working with a group at the University of Arizona called iPlant, which is really to develop cyber infrastructure for computers. And they have a large education module where they're specifically developing software. And they have generated something called the DNA subway, which we're going to be using in our class, which is very user-friendly. And so part of the problem is that it has to be written specifically for, you know, for classroom use, but it has to also be useful to discover things. And so people are, like I said, just starting to come to grips with these things. So I think computationally we can do a lot. Our lab, we figured out, is about $120 or $150 per student for the whole course, to do both the wet lab and the computational. So that's not, but you know, it's 25 students. You know, to scale it up, you'll need more TAs and things like that. But I would say we could probably scale it, I don't know, you'd have lots of sections in there, too. That's just, what's 3 hours twice a week. Right. Sure. Yes.
Would it be possible to infer how much evolutionary stress an organism had undergone based on the percentage of transposable elements in the genome?
I wish. We know so little. That's a really good question. That is a really good question. But we don't really understand the connection between stress and transposons. Okay, we don't know. Some respond differently. So I guess so what you're saying is sort of if an organism has a lot of transposons, does it mean that they've been stressed?
So like highly mobile organisms, like the grasses that can just blow their seeds along in the wind and be carried along by animals, they've got like up to 85–90%. Right. Meanwhile, more sedentary animals that have like a very specific habitat would have less because they're really specific and they would tend to die out if conditions change too much. So I was just trying to. It's
possible. I mean, that's the neat thing about this area is, all ideas are. That's a pretty good idea. Plants have bigger genomes because for some reason. I mean, their genomes are much more flexible. They form polyploids. Plant genomes, so I showed you the grasses. They differ in size by, 30-fold. I mean, all mammals basically have a genome that's within 2-fold. So animal genomes, mammalian genomes, are much more narrow. Reptiles have a large range in genome size. Like I said, we're just sort of finding this stuff out now, and so we're just characterizing it now, and the next step is to find out, does this mean something in terms of evolution or existing life or whatever? Right. Yep. Stay tuned, right.
Are there other questions?
I want to know on the transposable element, you were showing us a picture of petunia and the colored corn, that's a result of it. Is there any results you can see in a person? You can say, oh, that's caused from that? And the second thing, so I was wondering about your teaching methods, if those can go down to really the grade school. So
great questions. Okay, the first thing with humans is plants, because of the way they grow, they make sectors. So we can see the sectors. Animals, when they grow, their cells move around. So you generally don't see patches like you would in a plant. But there are human diseases that are caused by the insertion of a transposable element. So especially some of the hemophilias, that they're caused by an insertion because, people who have hemophilia, go to the doctor many times, and then they can figure out what the basis, and they know the genes involved, so they can sequence the genes and see what the cause of the mutation is. And in a few cases, they have found that there's actually a transposon that's stuck in the gene. So, yeah. And the second question, Jim Burnett has had a year class. He organized a year class at Athens Academy, which is in Athens, Georgia, for juniors in high school. Whether we can go, I really feel more comfortable sort of staying, with our comfort zone. I mean, what we can teach. I think when it gets down to younger students, we don't really have the teaching style. It's like I think it's a completely different experience. So, but I think that we could have middle school teachers, and I'm sure eventually we'll do this, take workshops in our class, and then they can adapt it to their class in a way that they think would work. Because I don't work with, I had middle school kids, children, but besides that, I don't know. So I think the best way with any of these is really to teach the teachers, and then have them translate it or deconstruct it for their age group, the age group that they're teaching. Right. Okay. Okay.
If you were to look at a really enormous gene pool like, say, Africa, where you have wide variability, taller, shorter, lighter, darker, speedy, not so, would you expect to find more of the trends?
Yeah. Actually, no. Probably not. Humans are, there's more genetic diversity in Africa, okay, because that's where humans started, and it's been the longest time. But no, most of that diversity is not due to transposable elements. But actually, we don't find elements move. Humans are really dull when it comes to transposable elements. We have very few. Our genomes are very, very stable, and most of it is because we really evolved from tribes that were maybe like 100,000 years ago. Our species was down to like 1,000 individuals. which means that we are all, I mean, all humans on this planet are really, really similar. You know, we don't have actually that much diversity compared to other mammals.
Well, we've worked Sue pretty hard.
It's all right. My pleasure.
Thank you very much.
Thank you.
I would like to